Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is part 3 of an oncology series presented by Clario entitled Integrating Objectively Measured Safety Endpoints and Patient-Reported Outcomes in Oncology Trials, a Scientific Approach. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the richest clinical evidence in the industry. Across decentralized, hybrid, and site-based clinical trials, Clario's deep scientific expertise, global scale, and the broadest endpoint technology platform allow their partners to transform lives. But now, without further ado, I'd like to hand the mic over to our moderator for today's event, Dr. Todd Rudo, EVP and Chief Medical Officer at Clario. Dr. Rudo, you may begin when you're ready.
Great, thanks, Ryan. And thanks to all of you who have taken the time to join us today. We're obviously really glad to see such enthusiasm and interest in this session, where we'll discuss endpoint collection in oncology trials with a focus specifically on cardiac safety, detection of neurologic toxicity, and also the role for patient-reported outcomes. So I'll take a moment here to introduce the panel joining me today for the discussion. As Ryan said, my name is Todd Rudo, and I'm the Chief Medical Officer at Clario. Dr. Vik Patel is our Vice President and CMO for cardiology at Clario. He's board certified in cardiovascular disease as well as electrophysiology. He earned his MD and PhD at the University of Colorado, completed his cardiology training at the University of Pennsylvania. In addition to his 12 years of academic practice, Dr. Patel has more recently focused his career in pharma development and joined our cardiac safety team here about 1 year ago now. Dr. Tanya Varselli is the Director of Scientific and Medical Affairs for Cardiovascular Imaging. She received her PhD in biomedical engineering from Case Western Reserve University, has over a dozen years of experience at Clario supporting imaging components of clinical trials across all phases, and her expertise spans multiple therapeutic areas. Dr. Kelly Jumai has 13 years of experience in behavioral and life science research, serves as Director of ECOA Science and Consulting at Clario. She has domain expertise in the implementation of electronic outcomes assessments and the development and validation of patient-reported outcomes to support regulatory submission. And today, Dr. Jumai will share an overview on the evolving role of patient-reported outcomes in oncology drug development. And then Dr. Adam Jagodinsky serves as the Principal Scientific Advisor and Oncology Lead for Clario's Precision Motion Group. He received a PhD from Auburn University with an emphasis in biomechanics and earned tenure at Illinois State University, where he conducted research on the mechanics and control of gait and posture, as well as integrated technologies for field-based human movement assessment. Go ahead to the next slide, please. So just briefly, here's our agenda for the next hour or so. Drs. Patel and Varselli will focus on cardio-oncology, specifically ECG, blood pressure, and cardiovascular imaging assessments. Dr. Jagodinsky will share with us a fairly novel technology for the detection of chemotherapy-induced peripheral neuropathy. And then finally, Dr. Jumai, as mentioned, will focus on the evolving role for patient-reported outcomes in oncology development. You can go to the next slide. So just to help set the stage for today's webinar, I wanted to highlight what makes oncology drug development such a unique space to work within. Many of the considerations and challenges listed here are unique to oncology and therefore represent a fairly substantial deviation from the more typical or established norms of drug development in other therapy areas. So just to highlight a few, in oncology development programs, rarely is a healthy volunteer phase included, nor is a placebo comparator arm present. So it makes data interpretation fairly challenging, especially when confounding factors are present, including significant comorbidities. Differentiating drug effects in that context from background disease-related effects can be quite difficult. The patients themselves are unique in that they're often very heavily pre-treated with prior cancer therapies, probably failing those prior regimens, and therefore fairly sick as they're entering these studies. They've also had side effects from those previous treatments, and those toxicities could carry over into this next trial as well. Adverse event rates are much higher in that setting, and so regardless of any treatment-related effects, again, confounding safety data interpretation, since there's really no placebo comparison, and therefore difficult to understand what may be causal in looking at those adverse events. Any proposals with consideration to add further assessments within a protocol need to be balanced against the consideration for increasing patient burden. And while that's true, of course, in any therapy area, it's particularly a concern when treating sick cancer patients in such a frail state. And last, I'll just call out that the drugs themselves, in some cases, they're far more toxic than those in other therapy areas, yet there's a higher tolerance for that toxicity from a benefit-risk perspective, as it's quite different. In the past 10, 15 years, however, you've all seen a shift towards more targeted therapies. These have reduced toxicities in general. And while this is advantageous and great news for patients, this shift introduces some operational challenges in that finding patients to participate in clinical trials, given the very narrow inclusion-exclusion criteria, is a difficulty for sponsors and sites. Sponsors are now seeking, therefore, only a small subset of patients with a particular cancer type, rather than sort of all comers, if you will. You can go to the next. So with that said, I will hand over now to Dr. Patel to let him take us through the key considerations when assessing cardiac safety in an oncology development program. And he will focus initially on ECG and blood pressure. Dr. Patel.
Thank you, Dr. Rudo. We can go to the next slide, please. So to begin with, I just want to mention, why do we even concern ourselves about looking at cardiac safety in drug development? Well, I think we know all drugs, regardless of their mode of delivery, will enter the bloodstream. And as such, they'll go through the heart, be delivered to the tissues. And exposure of these new assets to the heart has the potential for cardiotoxic side effects. So we have to assume that there is a cardiotoxic side effect until we can rule that out using clinical data. Let's go to the next slide, please. As Dr. Rudo mentioned, we really have to consider the benefit-risk ratio when we develop all new therapeutic agents. This applies also to oncologic therapies, but there is a difference in the benefit-risk balance with an oncologic agent, where the patients often have fatal disease and there are few therapeutic options. So the balance is shifting more benefit, and we're able to accept higher risks in this population for that benefit. Still, you can't accept too much risk to negate the benefits. So we have to make sure that these patients don't have sudden cardiac death or dying prematurely, or they're left with severe heart failure where they really might be still alive, but really still have a poor quality of life and gain very little benefit. Let's go to the next slide, please. The current oncologic division guidance is fairly straightforward and clear. There's really a requirement to just characterize sufficient ECG effects for all new oncologic agents. This is then to inform the prescriber about how to manage and assess the safety. What you're really looking for is large effects in either blood pressure or QC changes, as we'll mention. These will usually go into the label, and then they'll be available for prescribers to really select the right patient populations and assess and monitor these risks appropriately when the asset is marketed. We can go to the next slide. Now, with regard to assessing the QTc interval in phase 1/2 oncology trials, what we're really concerned about here is ruling out the risk of sudden cardiac death. All non-cardiac assets have the potential for inducing torsades points or fatal arrhythmias. However, these events are so rare that they really can't be detected in a standard clinical trial design, specifically a smaller phase 1/2 oncology trial. So what we're using is the QTc interval prolongation as a surrogate for this. QTc prolongation can be assessed readily and sensitively, even in a small oncology trial. As Dr. Rudo mentioned, oncologic studies are also different from most other therapy areas where you're not studying healthy volunteers in the first-in-human trial. You have oncologic patients, and so you won't have placebo controls or positive controls to give you the sensitivity or really rule out just how reliable the findings are. Still, the regulators accept this and have made exceptions for these trial designs. When assessing QTc interval, just like in other therapy areas, we really want to look at the change in QTc in regards to exposure of the asset. So you're looking to collect intense ECG PK pairs. This is done by using continuous ECG 12-lead holders. This can also allow us to collect triplicate ECG, so it can increase the sensitivity for assessing the QTc interval. You really need to consider a wide range of exposures here. You really want to get to an exposure that's up to the recommended phase 2 dose, or even up to maximum tolerated dose, to make sure that you can really exclude a QTc effect that would be deleterious. And the idea here is the goal is to develop as a primary endpoint the concentration QTc model to rule out large effects in QTc changes. These are ones that would be greater than 20 milliseconds, which would have a high risk of torsades de pointe, which is rarely available in these kind of study designs. We can go to the next slide, please. So with regards to assessing the QTc interval and getting proper results, we really need to consider about looking at centralization of ECGs and having cardiology overreads. We've looked at this in almost 300 oncology trials here at Clario that included 18,000 patients and over 270,000 ECGs that had both central ECG reads and machine reads. And when you look at the machine reads versus the central reads, we can go to the next slide. You'll see that the machine ECG read actually carries a higher number of false positive readings. This is not surprising. Most ECG algorithms are designed to overread intervals so that you don't miss any safety findings. But what it can do is it can actually exclude patients if the QTc is an entry criteria, or it can actually give you an overly adverse safety profile for the asset when it's really not necessary. But if you look at the machine ECG reads, when they increase from 450 milliseconds to 500 milliseconds, you see that the difference between the central overreads increases with increasing QTc interval. But more importantly, the percentage of false positive findings increases when the QTc increases from 450 to 500, from 30% at 450 to greater than 75% when the QTc is greater than 500 milliseconds. So what this really tells us is that just relying on the machine interpretations is really not going to be that helpful to the patients or the program. And really, we should consider ECG overreads and centralization in this case to get the best results. We can go to the next slide. Outside of the QTc interval, there are other cardiotoxic side effects that can be induced by oncologic agents. And we need to look at other modalities to monitor these. So the 12-lead ECG can tell us easily about morphology and intervals, but there are a rare incidence of infrequent heart block, tachyarrhythmias like atrial fibrillation, or ventricular fibrillation that a 12-lead ECG can't detect. We have to use continuous ECG monitoring with a Holter or patch monitor to detect these. Blood pressure changes and vascular effects from oncologic agents are also a common effect. These can be detected by clinic blood pressure, home blood pressure, ambulatory blood pressure. We're really looking for large changes in this effect, just like with the QTc in phase 1/2 oncology trials. And then finally, myocardial pericardial effects from these agents. Heart failure, LV dysfunction, or pericarditis is something that we can usually assess with imaging, and also use some biomarkers that can help detect these earlier. We'll talk a little bit about. You can go to the next slide. For blood pressure monitoring, there's really 3 modalities available for oncologic studies and for other therapy areas. Clinic blood pressure monitoring is probably the one that's going to be used the most in oncology trial to detect the changes that you'll see as these patients come in for the clinic visits. These come in scheduled visits, and we have automated and standardized procedures now available to reduce variability for this assessment. Home blood pressure is another modality used in oncologic trials. This is something that can reduce the trial burden on the patient and can increase the frequency with which you're getting blood pressure assessments. And then finally, ambulatory blood pressure is available. This can record 24-hour blood pressure, both in the daytime and nighttime, to see if there's an effect on circadian rhythms. This is something you're going to use less often in an oncology trial where the therapies are given intermittently. But it is still available. We can go to the next slide. With regards to kind of cardiotoxic effects we would see with an oncologic agent, I'm going to just highlight the tyrosine kinase inhibitors. This is a class of drug that's commonly used and has a representative cardiotoxic profile, I think, of a lot of oncologic agents. Many of the TKIs are small molecules, so they'll circulate in the heart and other tissues, although now there are more that are monoclonal antibodies. And it'll inhibit vascular endothelial growth factor as well as other kinases. Tyrosine kinase inhibitors, tyrosine kinases are expressed in the myocardium and the vascular tissue as well. So this is why they might have effects on both the heart and the vasculature. And the kind of toxic effects we'll see are typical of what you see with a lot of oncologic agents. There's hypertension, LV dysfunction and heart failure, QTc prolongation, arterial thrombosis, and myocardial infarction and ischemia. Let's go to the next slide, please. And so finally, I just want to give an overview of what we think might be best practices for monitoring for cardiac safety in oncologic study. We've discussed getting frequent and enough ECG assessments in all phases of development. This is something that's simple and easy to do, and I think that most sponsors follow well. You can really consider, I think, getting biomarker assessments. High sensitivity troponin biomarker and BNP are both readily available, inexpensive, and easy to assess. This is something that can really help detect myocardial damage earlier on before imaging or other modalities can, and it may actually help prevent permanent LV dysfunction or damage. Blood pressure assessments, again, we talked about using really clinic BP and home BP are more the foundation in oncologic studies, although ambulatory blood pressure monitoring is available. And you're really looking for large changes in blood pressure with these. And then finally, serial assessments by cardiac imaging for LV function, typically done by echocardiography or MRI, but you can also use nuclear imaging for these. And this is something that Dr. Vericelli will talk about in more detail next. So we can go to the next slide, and I think Dr. Vericelli will start here.
Hi everyone, thanks Dr. Patel. So I'm here today to talk about cardiovascular imaging and oncology trials. So let's dive right in and move to the next slide. So there are several national and international organizations that have issued cardio-oncology guidelines. The ones that I'm referencing in my presentation today are the ESC guidelines and the ICOS consensus statement from 2022. So we can move to the next one. So there's 5 main common cardiac complications and toxicities. And so Vic already did a great job talking about arrhythmias and QT prolongation and hypertension. And I'm going to talk on my portion today about cardiac dysfunction and myocarditis, because those are the 2 that utilize cardiovascular imaging. One of the overarching themes of the guidelines is that for a given patient, the absolute risk for cancer therapy-related cardiovascular toxicities is going to depend both on their baseline risk and changes with exposure to cardiotoxic therapies over time. So how sick is the patient before treatment? What known toxicities does the drug have? And the clinician or the investigator is evaluating that risk according to the likelihood of its occurrence for that patient and the severity of that complication. And the overarching aim is to personalize approaches to minimize cardiovascular toxicities and improve both cancer and cardiac outcomes. So let's move to the next slide and talk about those mechanisms of cardiac injury and toxicity. So I have a table here. I'm not going to walk through it, but we have some example therapies and different mechanisms of damage. But the overarching principle is that there still remains overlap between cancer and healthy cells that can be damaged or targeted for programmed cell death by these cancer therapies. The primary concern is endomyocardial damage of the left ventricle that can lead to heart failure, but the middle layer of the heart can also be impacted with myocarditis, and the outer layer with pericardial inflammation and effusion. And it's important to note as well that combination therapy will increase that cardiotoxicity risk to the patient. And on the next slide, we'll look at cancer therapy-related cardiac dysfunction. So how is that being, that's one of the main common toxicities we see. How is that being assessed? And the primary way to do that is through the measurement of the left ventricular ejection fraction, or LVEF. LVEF is simply the fraction of the blood ejected in a single cardiac cycle. So what % of the blood is being injected in each heartbeat? On average, the rule of thumb, while this varies by gender, is between 50 and 70% is normal. And on the left-hand side, you can see normal LV systolic function pre-chemotherapy in that heart. But then on the right-hand side, post-chemotherapy, we see severely reduced systolic function, a decrease in LVEF. That heart is dilated, it's pumping poorly, and there's a smaller contraction of the muscle. And so clinically, this can end up leading to heart failure. So how are we going to monitor this over time? What cardiac imaging modalities are preferred? The primary method is transthoracic echocardiography, or just echo. It's the preferred modality given the type and the quantity of assessment. So you can look at the structure, the function of the heart, systolic and diastolic function. It's also logistically less expensive. It's globally available. It does not have radiation exposure. Usually you don't have to even give contrast. It's non-invasive. So it's actually convenient as well. The primary method for LVEF assessment is preferred option is via 3D assessment, but it's primarily globally still done with 2D and the biplane-Simpson's method for LVEF. In patients that have inadequate image quality, where 2 or more of the left ventricular segments are not well visualized, it's recommended to utilize contrast. And if you utilize contrast, you want to do it consistently to minimize that variability of the LVEF assessment. Your second line option is cardiac magnetic resonance, or CMR. This is used in subject with poor quality echo windows in evaluation for cardiac dysfunction. However, it is the primary method that's being used for the evaluation of myocarditis. And then last is MUGA. We really try to avoid that whenever possible. This is the last line item. It has radiation exposure and a really limited assessment of just LVEF. And of course, in clinical trials, the technique that you want to use needs to be available across all of your sites with local expertise in applying that methodology. And the same imaging modality needs to be used throughout the entire study to eliminate inter-technique variability. So on the next slide, we'll look at the assessments that should be made at baseline. This is a graphic from the ESC guidelines. There's a lot of abbreviations. We're not going to go into all of it. Suffice it to say that the take-home message from this is that you want to look comprehensively across the heart at baseline. So the left ventricle, the left atrium, the right ventricle, the right atrium, valvular assessments, pericardial diseases. You want to look at the whole thing to really get an idea of how to stratify that patient's baseline risk. 2 key assessments are LVEF and global longitudinal strain, which is abbreviated as GLS. And those baseline values are really going to help you stratify that baseline risk, but also importantly, to serve as comparators for that subject longitudinally over time. For clinical trials, it's really important to carefully consider your eligibility criteria relating to cardiac function. Ideally, you'd like a relatively homogeneous risk profile for your patients that are enrolling at baseline. And of course, you want to include a conservative image monitoring plan while on treatment and during long-term follow-up. On the next slide, we'll evaluate GLS in a little bit more detail, so you can see the video there. GLS uses speckle tracking over the cardiac cycle. So the motion of the heart, people think of it as just moving in and out, but it's actually more akin to wringing out a wet rag. So you're squeezing it, you're moving it in multiple directions, and that's what strain is tracking, is that movement of the endocardial border of the left ventricle. And the importance of the strain measurement is that you can recognize early preclinical changes to the motion of the heart before there's a decrease in LVEF. So strain is giving you an early window into potential dysfunction from cancer therapies. And on the next slide, we'll see that because of that, GLS has also been introduced and recommended in the current oncology guidelines, as well as LVEF. So this is an image from the guidelines showing the grading of cardiac dysfunction. The pink box that I'll draw your attention to is those that are using cardiovascular imaging. So you'll see both LVEF and GLS. You're looking at absolute thresholds, but importantly, you're also looking at changes from baseline over time. And again, this illustrates the importance of consistent, good quality imaging at both baseline and follow-up. There's a lot of variability in echo imaging, in all cardiovascular imaging. Some sources of variability, you can have different acquisition protocols, different view sets that are required, different methods of measurement, different readers making those assessments, and then just the natural variability of where you place the electronic calipers to make the measurement. So you want to minimize as much as possible this variability. And so during clinical trial design and startup, you want to focus on providing a standardized view set and an acquisition protocol that should be used across all sites for every scan for that patient. And then, of course, the value in central reading is in using a consistent measurement methodology by a single reader across all sites and all subjects longitudinally. And decreasing that variability is important for the investigator to feel confident applying these criteria to grade cardiac dysfunction, which could impact the treatment paradigm for that patient. The frequency of this imaging monitoring that you're doing for these patients that are on treatment and for follow-up really depends on the cardiotoxic profile of the drug being utilized, as well as your enrolling patient's baseline risk. And of course, if you have a subject that presents with new cardiac symptoms on therapy, you want to do some ad hoc imaging as well. On the next slide, we'll turn and talk about myocarditis. That's the inflammation of the myocardial heart muscle. You'll see here is another graphic from the guidelines. The pink boxes, again, are where they utilize cardiac imaging. And in this case, it's CMR is the preferred imaging for myocarditis. So you'll see the major criterion for diagnosis is actually via CMR using the modified Lake Louise criteria. And on the next slide, we'll dive into the Lake Louise criteria a little bit more. So there was an update in 2018 to the Lake Louise criteria. The requirement now is that you have to meet both criterion in order to be able to diagnose acute myocardial inflammation. So the first is you need a T2-based marker for myocardial edema, and the second is a T1-based marker for associated myocardial injury. And you use several different sequences to do that. So you're requiring T2 mapping or T2 weighted images, T1 mapping, ECV, LGE assessments. You're also looking at supportive criteria for pericarditis or left ventricular dysfunction with different wall motion abnormalities. So you're using all of those sequences in a CMR to make this assessment. And then on the last slide, we're just going to kind of wrap it up. The key item that we want to talk about is detecting myocardial toxicity early in these clinical trials. So the first thing you'll see is myocardial stiffness, so a little bit of diastolic dysfunction. And then you'll move into impaired global longitudinal strain. And finally, when the heart can't compensate as well, you'll see a reduction in LVEF. So not as much blood will be output from the heart with each beat. And the further you get down the line towards heart failure, the less reversible this myocardial injury may be. And so as Vic and Todd were saying, it's really important to have all of the information from the cardiovascular imaging and the ECG so that that investigator can make an informed risk-benefit assessment for that patient. They may have to change treatment or reduce a dose, or they may just keep monitoring more frequently. And so having that information is really important during the course of these studies. And so now I'll hand it over to Adam, and he's going to talk a little bit about another issue, which is drug-related peripheral neuropathy. Thanks.
Thanks, Dr. Verselli. So today I'm going to be discussing novel solutions for detection of drug-related peripheral neuropathy. Next slide, please. So to start, I would like to give some background to help us navigate the topic. So chemotherapy-induced peripheral neuropathy, or CIPN, is a persistent side effect of cancer treatment that impairs physical functioning, increases fall risk, and reduces quality of life in cancer survivors, as well as patients who are undergoing treatment. CIPN can result from multiple classes of neurotoxic chemotherapy drugs, including taxanes, platinum derivatives, vinca alkaloids, and there's an estimated prevalence reported between 25 to 90%. The etiology and natural history of CIPN varies between patients and class of chemotherapeutic agent. However, sensory and motor symptoms of CIPN are known to occur in the hands or feet, which is indicated on the diagram on the right. So this illustrates the effect of various classes of chemotherapeutic agents on structures of the peripheral nervous system, including nerve endings, blood vessels, the spinal ganglia, mitochondria, ion channels, and myelin. Next slide, please. So here we show how CIPN affects sensory pathways that control balance. And specifically on the far left portion of the flow chart, we see three main sources of sensory input that regulate balance that include vestibular, visual, and proprioceptive. And so I would like to draw your attention to the bottom box that highlights proprioceptive input, and that relates to peripheral sensory structures that detect movement. So the proprioceptive input is integrated with other sensory input in the brain and the brainstem, and further regulates motor impulses that control eye movement and postural adjustments that are really essential for maintaining balance in day-to-day tasks. SIPN affects the peripheral sensory structures and alters that integration of proprioceptive input, and that can negatively impact balance and mobility. So for example, on the right, we see two graphs representative of postural sway patterns for a healthy control on the top and a patient with SIPN on the bottom. The postural sway patterns show distinct differences in both forward and backward body sway, which is shown on the vertical axis, and side-to-side body sway on the horizontal axis. And this indicates impairment in the balance control strategy. In addition, on the right-hand side, we see box plots that compare two aspects of walking gait between young and old healthy controls, which are shown in blue. Young and old prostate cancer survivors, shown in gray. On the left-hand sample includes patients who received past therapeutic intervention, and the right sample showing sample that are ongoing, having ongoing therapeutic intervention. And the red dots show a sample of very old healthy controls. So the top graph shows that cancer survivors who received therapeutic intervention tend to spend more time in double support phase. So that's when 2 feet are supporting the body during walking compared to healthy controls. And this type of compensatory pattern is visible in very old populations who spend more time in double support to preserve balance and mitigate fall risk. So the bottom graph shows that compared to controls, survivors who received therapeutic intervention exhibit a smaller foot angle when pushing off during gait, tending toward that of very old control subjects. So this measure is indicative of a reserved gait strategy that may be adopted to compensate for motor dysfunction and also instability during single limb support of gait. So ultimately, these examples highlight some of the functional impairments that may exist in association with chemotherapy-induced peripheral neuropathy, and that can be feasibly measured with wearable sensors in a clinical trial setting. Next slide, please. So this slide highlights data from a 2017 study that looked at the relationship between patient-reported neuropathy symptoms and digital measures of postural sway in 28 of 33 women that were enrolled with early-stage breast cancer during chemotherapy treatment. The graph on the left demonstrates changes in patient-reported sensory symptoms, which are measured by the SIPN-20 sensory subscale, shown by the dotted line, and also changes in objective postural sway measured, shown in the solid line, over the course of 3 cycles of chemo treatment. So a decrease in the dotted line represents a worsening of patient-reported sensory symptoms, while an increase in the solid line represents a worsening of the objective balance measure, or postural sway. So looking specifically at balance here, the data showed a 28% increase in postural sway measured from baseline to the completion of the first cycle of treatment, and also a maximum percent increase of 43% in postural sway from baseline. So looking at the scatter plot on the right, this shows a significant negative correlation between patient-reported symptoms, the SIPN-20 sensory subscale, and objective measures of postural sway between 1 to 3 months post-treatment. So there's an association there between the patient-reported outcome and the objective balance scale. So the next slide shows some exploratory analyses from the same study, highlighting response trends of objective balance measures and patient-reported sensory symptoms associated with specific treatment characteristics. So the top row shows response to prior doxorubicin and cyclophosphamide exposure. The middle row shows patients' response to pegfilgrastim, and the bottom row shows response to weekly paclitaxel. So the left column shows objective balance outcomes measured by the root mean square excursion, which is basically the amount of postural sway deviation from the mean. And in the right-hand column, that shows patient responses on the EORTC SIPN-20 sensory subscale, which consists of 9 sensory items rated on a 4-point scale. So both the objective balance and patient-reported SIPN symptoms show trends of worsening throughout the duration of each characteristic treatment and at 1 to 3 months post-treatment. The next slide provides an overview of Clario's solutions that are currently being implemented on an ongoing clinical observational study to assess adverse effects associated with SIPN. Thank you. And to preface this, the Opal wearable sensors and mobility lab software have a robust history of scientific validation and also use in clinical cohorts, and can be implemented in clinical trials to objectively assess movement and function in conjunction with standard patient-reported outcomes. So performance measures include instrumented tests across 3 main domains that are gait, balance, and mobility. And so you can see for gait, we see common clinical assessments such as the 2-minute walk, the 6-minute walk. We see for balance, there are a number of different conditions in which balance can be assessed. So that could be removing vision or changing the surface to alter different sensory inputs. And the third is mobility, such as classical tests like the tug and the sit to stand test. All right, so moving to the next slide, we see that data collection timeline. This is a data collection timeline for early detection and monitoring of SIPN adverse effects using Opal instrumented in-clinic assessments of gait, balance, and mobility performance measures, along with objective neuropathy measures, patient-reported outcomes, and daily activity mobility assessment. So for this study, patients will be assessed in 4 to 6 week intervals during treatment for detection of SIPN adverse effects, and every 3 months during a 1-year follow-up period for long-term monitoring of SIPN symptoms. And on my last slide, I have just a few takeaways. So SIPN is an adverse effect of common cancer treatments that affects patients' quality of life. Balance and gait impairments are associated with SIPN and are detectable through treatment and in long-term assessment of cancer survivors. And these are important measures as they lead to falls and increased fall risk and are meaningful to patients. And finally, instrumented assessment of balance and gait can provide objective insight into adverse effects due to neurotoxic chemotherapy treatments. And with that, I will hand it over to my colleague, Dr. Kelly Dumais, for her topic on the role of patient-reported outcomes in oncology.
Thanks, Adam. I'm happy to be here again. Yeah, I'm a director in our ECOA science team. So our science team provides guidance on clinical outcome assessment strategy, and I'm happy to talk to you today about patient-reported outcomes in oncology, or PROs, and oncology trials. Before we move on to the next slide, I was going to say I'll just provide a bit more of a background as well on the importance of PROs. So what we've seen, I think, overall in the industry is that the use of clinical outcome assessments or COAs in oncology trials has been historically less than in non-oncology trials. And the use of PROs has been historically low because the main objectives, of course, of oncology trials are focused on survival and tumor responses, and rightfully so. And when it comes to safety and adverse events, this is typically assessed using objective methods like we've heard about EEGs and also clinician reports, and again, rightfully so. But now we're seeing this growing importance of also collecting the patient experience from the patient's perspective, as objective methods alone may not provide that full picture of therapeutic benefit or risk-benefit evaluation. So we are seeing this growing interest in patient-reported outcomes, and I believe we're seeing this for multiple different reasons. So now if you can move on to the next slide. The first reason we're seeing this, growing interest in oncology PROs is maybe obvious. It's the patients. It's that patients should and want to know how they will feel on treatment. So cancer survival rates are increasing, which is great with what this graph is showing. But that also means that more people now are living with cancer than ever before. So for example, in 2020, an estimated 17 million people were living with cancer in the US alone. So it's not just about survival, it's about how patients are living with cancer. And we owe it to patients to take their symptoms and experiences seriously, therefore measure their symptoms in a systematic way, right? And then be able to tell them with data how they'll feel when taking an approved treatment so that they can make informed decisions about their treatment in a world where their options are increasing. Which then brings me to the second argument on the next slide, that why we're seeing this increased interest in PROs is that competitive market landscape. So if we move to the next slide, I don't know if it's stuck. There we go. Thanks. So this is showing that there were 573 oncology indication approvals granted between 2000 and 2022. And we're really seeing that this rate is increasing drastically over time, such that when you see the mean annual approvals from 2000 to 2004 were 7 oncology drug approvals per year to 56 per year in more recent years, from 2017 to 2022. So that's a over 700% increase in oncology drug approvals. So when you think of all of the options that patients have, it's important to have your drug differentiate from those and stand out. And patient-reported outcomes are one way to be able to do that. And we're seeing them patient-reported outcome data increasingly put onto even oncology drug labels, so that patients can have, as I mentioned, a better informed information on how they will feel on treatment, and they want to know this information. So having a treatment that also gives data on the impact of quality of life and symptoms can be one way to differentiate your drug as well. And then the last reason, if you go to the next slide, and I think why we're seeing this increased interest in patient-reported outcomes is, of course, the regulatory landscape, right, and the regulatory interest. So here's a picture of the oncology regulatory landscape in recent years, and what you can clearly see is the increased attention on PROs from both the FDA and the EMA. There are lots of other oncology guidances not included here. This is just a snapshot of those that relate to PROs specifically, and you can see both the FDA and EMA updated their main oncology guidance in recent years, in the last few years. And in those updates, they've both emphasized the importance of PROs in better addressing the risk-benefit evaluation of oncology treatments. The EMA and FDA also have PRO-specific guidances for oncology, and I'll go into more detail on the FDA one in a minute. And FDA even also has annual public workshops, dedicated to COAs and cancer clinical trials, where they discuss different topics related to best practices in PRO collection. So we really are seeing this increased interest from the regulators on PROs. So if you go to the next slide, just briefly, I'll talk a little bit more specifically on what we should be collecting, right? When we say PROs are important, what is the FDA looking for? If you go to the next slide, it might be a little lagging. So there are 5 core PRO categories that the FDA have identified as being important in their most recent guidance that they released in draft form in 2021. And those 5 core categories are listed here. So disease-related symptoms, that means specific to the indication that you're studying. So there's lots of general quality of life assessments like the QLQC30, and that's important too. That will facilitate the assessment of things like physical function and role function. But you also need that indication-specific module as well to address the disease-related symptoms. Symptomatic adverse events. This could be addressed via the use of the PRO CTCAE, which I'll talk about also in a couple of minutes, and then also an overall side effect impact summary score. So they've really nicely outlined for you the 5 core PRO categories that they're looking for. And then I think one of the other biggest takeaways from this guidance that the FDA released is the recommendations on frequency of collection. So traditionally, PROs, when they're included in a study, maybe are collected at site visits only. maybe the first day is some of the early treatment cycles. But the problem with that is that these PROs typically have a 7-day recall period. So that means if you only collect the pro at the start of a treatment cycle at the visit, you're only collecting the patient experience data from the last 7 days of the previous treatment cycle. So you're missing a big piece of the picture there. So to collect a more complete picture of how the patient is feeling, you need more frequent data collection, potentially weekly. And because of that, we're seeing also kind of a shift in data collection mode towards at-home collection. So to accommodate that weekly assessment versus only at the clinic. So we are seeing that a bit more from our sponsors. If you go to the next slide, another big guidance that came out related to PROs, this isn't a PRO specific guidance, but I wanted to call it out here given the emphasis of this webinar also on safety and tolerability. A couple of months ago, the FDA released this guidance, and it really just calls out the use of PROs for the assessment of tolerability. The guidance states, patient-reported outcomes can provide a systematic and quantitative assessment of expected symptomatic adverse events and their impact on function to enhance the assessment of tolerability. So I think that's what's really important. It's that PROs are providing that systematic and quantitative assessment so that we can make sure we're systematically quantifying the impact of treatment on a patient's health. So, having those objective measures, again, undoubtedly important, but they don't say anything about how it makes the patient feel, right, or how it impacts their ability to work or do daily activities. So that's additional information that you're able to gather using PROs. And then again, that enhancement of the assessment of tolerability. We're not looking for PROs to replace any other method of safety and tolerability assessment, right? It's a way to just provide deeper insights into how tolerable a drug is from that patient's perspective. So if you go to the next slide, just to speak a couple of minutes on the, again, how are we measuring tolerability using patient-reported outcomes? And there are a number of maybe one-item measures that ask, for example, how bothered are you by the side effects of treatment? But then there's also this patient-reported outcome version of the common terminology criteria for adverse events. That's a mouthful. It's called the PRO-CTCAE. And this is a system, pro-measurement system developed to evaluate symptomatic toxicity in patients in cancer trials. It's an item library that includes 124 different items representing 78 symptomatic toxicities. And it's important to note that this is, again, used in conjunction with the clinician-reported CTCAE for the purpose, again, of that enhanced tolerability assessment. And it doesn't replace that clinician CTCAE, but it gives additional information on better understanding these types of symptoms that are actually subjective symptoms and are best known by the patient and actually measuring them from the patient's perspective. So in the next slide, you see actual data to show the importance of gathering this information from the patient themselves. So even when it comes to symptomatic adverse events that are best known by patients, the current practice is that clinicians collect this information through, their interactions with the patients. But these, research has shown, as you see here, that these impressions are not always accurate. So that the first graph here shows that patient-reported symptoms in orange and the same symptoms that were clinician-reported in blue, and it shows that those self-reports are more sensitive actually to those underlying changes in patients' functional status and actually identify symptoms earlier during the course of treatment. So clinicians are actually systematically downgrading the severity of symptoms. And on the right, you're seeing that the patient reported symptoms actually has better concordance with their overall health status than the clinician reported version. So just again, evidence that it's important to get these this type of information from the patient themselves because they really know their symptoms best. On the next slide, again, is just kind of a summary of why it's important to use these PROs to evaluate tolerability. You're getting more precise data by asking patients themselves. It also could reduce bias, given that there could be some bias in an interview with the patient-doctor relationship that could lead to bias. You're getting more sensitive and accurate data, and it's also potentially easier to collect self-report symptoms because you don't need the clinician to call, or you don't need that site visit to get patient-reported data. And it's also important, if you go to the next slide, to incorporate PROs early in the drug development process as well. So the FDA representatives have said the earlier, the better for incorporating PROs for a number of reasons. If you go to the next slide, you know, it's a way to optimize phase 3 PRO strategy. It's a way to make sure that you're submission ready, because the expectations from FDA is that they want to see pro data, and many drug approvals are based on non-phase 3 earlier phase drug trials. So it's important to just incorporate the PROs early in drug development. And then finally, you know, again, incorporating pros early to allow you to have that enhanced assessment of tolerability. If you want to jump to my last slide, I'm not sure if everyone's seeing a little bit of a delay. If you go one more slide to the options for pro, there we go. I'll end here. So we've established the importance of incorporating PROs, but again, how do you collect them? So of course, the most traditional data collection mode is paper, but we're seeing really a major shift away from paper and towards electronic data collection for a lot of reasons: better quality data, less missing data, et cetera. It's even recommended by the FDA Core Pro guidance to use electronic methods as well. So we have a provision device model, a BYOD, which is bring your own device, where patients use an app on their own phone and tablet, home computer, et cetera. And as mentioned, you know, another advantage of that electronic collection is that it allows for easy at-home data collection. So if you remember from the regulatory summary, with the FDA recommending more frequent collection of pros, you know, so say using a handheld or BYOD at home might be an easier way to collect those symptoms, such that you're not making the patient go to the visit for at-site completion, that you're allowing them to complete these patient-reported outcomes in the comfort of their own home using, you know, a BYOD, a provision device model. And the graph on the right shows that you're able to get superior measurement of quality of life when you do have that more frequent collection versus only at the site visit. So I'll end there, and I want to thank you for listening to all of our talks. I know it's a lot of information, so hopefully you got some great tips from our talk, and I'll hand it over to Ryan for the question and answer portion. Thank you.
Well, thank you very much for that very insightful presentation. I would like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, we've already received some great questions so far from the audience, so I think we'll get ourselves started with those. Dr. Rudo, you might have some already.
Yep, great. Thanks, Ryan. Well, I'm thrilled to see so many questions have come in throughout the session. Invite the panelists. Looks like they have to turn on their video and unmute, and you know, we'll do our best to get to as many as we can right now, but certainly we'll follow up direct by email to anyone whose question is not answered real time here this morning. So with that said, first question here is for Dr. Verselli, and this individual asks, you mentioned that 3D echo is better for assessing ejection fraction than regular two-dimensional echo. Is 3D echo a standard now, and would regulators expect that we do this in our studies, or is 2D echo still the better choice?
That's a great question, thank you. So I do think 3D is eventually going to be the standard. It's already the primary method recommended in the cardio-oncology guidelines. Just to take a step back though, so a 3D echo basically uses a specialized probe with transducer elements that are organized in a matrix formation instead of a single line, and that captures 3D images of the heart and the structure. So the advantage of using the 3D for LVEF and volume measurements is that 3D doesn't rely on any geometric assumptions of the shape of the heart the way that the 2D assessments do. And it also eliminates a common source of error in 2D measurement, which is the foreshortening of the left ventricle that you'll see in some views if they're not acquired perfectly. 3D is, however, not without challenges, right? So it's newer, it's not widely globally utilized yet. And of course, it has the same challenges with most of imaging. In order to get accurate measurement of the volumes in the LVEF, it's image quality dependent. And so you need sonographers that have experience in acquiring good quality 3D views. So I think eventually it'll move there, but right now, almost all of our studies are still using 2D biplane symptoms assessment.
Okay, great, thanks. This next one is for Dr. Jagodinski. What is the current regulatory landscape surrounding the use of wearable technologies for gait and balance assessment in clinical trials?
Yeah, thanks, Todd. So currently, most digital measures of gait and balance are included as exploratory endpoints in clinical trials. However, there's an industry-wide movement to increase awareness of digital health technologies and digital endpoints for use in clinical trials. And across the industry, there's an open and ongoing conversation with regulatory bodies really meant to shed light on and facilitate the process for regulatory approval of novel endpoints, novel digital endpoints. So overall, this is an emerging area in a clinical trial space, and you know, looking forward to having more regulatory acceptance of these measures.
Great, thanks, Adam. Okay, so for Dr. Dumai, this is a little bit of a longer intro. It makes intuitive sense to look at more than just overall survival or reduction in tumor size, but to also incorporate patients' quality of life. Striking the balance of not asking too much of a patient, though, by adding multiple PROs and potentially overburdening them with surveys is a concern. So as a two-part question, first one is, have you looked into patient satisfaction and compliance rates with electronic surveys in oncology populations specifically? That's a good question. And part two, do patients want to take assessments at home, for example, or would it be easier for them to just do at clinic visits?
Yeah, it's a big question. So yeah, studies have shown that oncology patients do want to report their symptoms, so they find it important and they do seem motivated to do so, and we do see that in the compliance rates that we do get. So we have been able to look at the compliance of electronic PROs, and we do see that they are motivated to do so, and we see a high compliance there. I agree that we don't want to throw every quality of life assessment at them. You know, we do need to strike that balance because these are, you know, potentially very sick patients. So it's important to balance that, and we want to be thoughtful in our PRO strategy, you know, and only include those PROs that are important, and consider, you know, options that are easier for them or more conducive to their lifestyle. As I mentioned, you know, potentially allowing them to complete them at home. As you say, you know, some maybe want to just complete it at the site. So I think optionality and flexibility is what's important, and just providing those options and flexibility, and an electronic implementation can be an easy way to do that, to allow them to do it at home or at the clinic.
Great. Okay, thanks. Yeah, that's a good question. Time probably for one more here. We'll take one for Dr. Patel. What are some of the operational challenges you've commonly seen in assessing QT or cardiac safety specific to oncology studies, and how do you see those best handled?
Yeah, thanks. That's a very good question, and this can be very difficult with a QT study when you're doing concentration QTC effect to try to collect ECGs. You're trying to get PK from the patient, you're trying to do blood pressure and other assessments all at the same time, and then get ECGs as well. It's a lot of burden on the site staff. So one of the things that helps is to actually collect the ECGs using a continuous 12-lead ECG. You can put the 12 leads on the patient, they can wear it for the whole day. The leads don't move, so they're always consistent. And then the continuous ECGs, the site doesn't really have to worry about it. They'll be extracted later, they'll be matched to the PK time points. You can even go back and get points that may not have been collected during the study if necessary. So that's one thing that can really help significantly, I think, reduce the burden on the site and the patient during these intensive PK days.
Great, thanks, Vic. All right, well, thanks again to our audience for joining us for all this engagement during the Q&A segment. As I mentioned, for any of you who asked questions we didn't get to just now, you'll receive direct follow-up either from me or one of our panelists. With that, I'd like to thank each of our four panelists for their really interesting and informative presentations, and I'll turn back over to Ryan to close us out.
Yes, thank you, all of you, very much for those answers to those questions, as well as that wonderful presentation. We have reached the end of our time here today. As you heard, if we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have some further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar and the entire three-part Clario Oncology series. Remember to visit xtalks.com for more Clario webinars coming in the new year. For now, you will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you.
Thank you.
Right.